Welcome to the Faculty Focus podcast, live from our annual Faculty Research Day with your hosts
Marleen
and
Teun,
highlighting the research of our professors and PhD researchers. Welcome back. Here with us now we have PhD researcher Rosanne Verboven. Her topic of today is low-grade mucosal inflammation in aged SAMP8 mice blunts recovery after DSS-induced colitis. That's quite a mouthful. Welcome, Rosanne. Can you briefly introduce yourself to our listeners?
Hello, my name is Rosanne. I studied Biomedical Sciences with a major in neurosciences and a minor in research. After my graduation, I first worked a year as a project coordinator in a CRO company in Leuven. And after that, I started my mandate assistant role here at the University of Antwerp in the lab of cell biology and histology.
All right. Well, thank you for being here, Rosanne. The first question is a very broad question. Imagine you run into a friend that you haven't seen for ages and she has no feeling with science whatsoever. How would you explain your research to her?
So what I'm studying or investigating is whether the connection between your gut or your gastrointestinal system and the brain, whether it plays a role in the disease initiation and or progression in Alzheimer's disease.
And you use SAMP8 mice for that? What type of mice are there?
So in the 70s, these are actually just wild-type mice, and in the 70s, they noticed that part of the littermates showed accelerated aging. So there's no genetic background known about these mice, why they're aging faster. Mm-hmm. And then from the 70s on, they specifically crossed these, or breed with these mice to sustain that phenotype. So there are actually different SAMP lines. So this one is the 8 line, and every SAMP line has its own characteristics in where they are faster in aging. And then you have the littermates that were just normally aging. They are called the SAMR1 mice from then on.
Okay, so it helps with speeding up the research because they age quicker.
Yeah, so the reason why I use it in my specific context is because in Alzheimer, actually the most patients, or like 95% of the patients of Alzheimer, have no known genetic background why they develop Alzheimer. And on that sense, it was interesting to use this model rather than a genetic engineered model to study some aspects of Alzheimer's disease.
So your research links also the GI dysfunction, so the gastrointestinal dysfunction, to faster aging. Can you explain a bit this hypothesis a bit more?
The most studies that are done are not like conclusive, let's say, so it's more like retrospective studies that they did. But for example, there's a study that saw that like in a worldwide study that people with inflammatory bowel disease have a slightly higher risk in developing dementia or neurodegeneration in later of life. So therefore they wondered like how come, and also a lot of patients with Alzheimer, but also with Parkinson, for example, are also struggling with gastrointestinal symptoms. So it's a bit like, is the gastrointestinal symptoms before or after? Is it so? That's a bit what still needs to be.
Egg,
that still needs to be unraveled. For Parkinson, there are more, clues that it can really start in the gut and then go.
Oh, that's new.
Go into the brain via the gut-brain axis. So that's already known that if you, for example, inject amyloid, which is the family of where the pathogenic enzymes, alpha-synuclein and amyloid beta belongs to. If you inject amyloids in the gut, they can travel through the brain and start pathology there. So that's already known. But it's always often done in genetically models that already have this pathology. So it's a bit, does this also goes for wild-type mice or not? So some studies say yes, some not. So it's a bit still, discutable, let's say,
of course,
whether or not it's possible.
Yeah, it's a fascinating link anyways.
Yes, so indeed in general, investigation in the gut-brain axis in the recent years, also in autism or whatever, you see a lot of more papers popping up, the connection between it. So therefore also with Alzheimer, where I specifically look into that.
So I've noticed in your slides that you say that the concept of leaky gut syndrome has been challenged recently. So this leaky gut syndrome, can you explain it a bit more and why it's been challenged?
So the leaky gut in se is not challenged. It's the aspect that aging alone can induce the leaky gut that has been contraindicated in literature. So first, what is a leaky gut? When you age, you have your barrier and you have proteins that hold your barrier intact. In the gut, and with aging, these proteins that keep them together, let's say, the cells, if they come leaker over time or less connected, let's say.
Yeah, there's space between the cells.
So and therefore substances that normally cannot pass through the barrier are now able to pass through to the barrier. So then you have inflammation. Yeah. So there is already known that some kind of like stress, inflammation, and different kind of pathologies can induce that your gut become leakier. So that there are more substances coming through. It was also always thought that it was the case for aging. However, a recent paper saw that in young human patients, young and old, and comparable with IBS patients, that actually the barrier remains constant over time. So that age does not alone, let's say, could not make the barrier more permeable in say. So that's why we wanted to check this with mice as well.
And the IBS patients is an abbreviation for?
Inflammatory bowel syndrome.
Okay.
Sorry.
Yeah, no worries.
Inflammatory bowel syndrome.
Just a question indeed. All right. So it's becoming very clear that the gut is very important actually for our health. Do you know about ways to prevent aging by helping our gut? Was it something that came up during your research or literature search somewhere?
What is known is actually that like the Western diet is not the healthiest diet, let's say, so that if you eat more like the Mediterranean diet, that's actually better for your gut. But what that has influence for the rest, I don't know.
No worries. Well, a bit off topic, but if you're in the lab, can you describe your dream day in the lab? What would make it absolutely perfect?
So I'm also a mandate assistant, so I also teach. So that the teaching that I do is always half a day, so it's not just 1 hour of teaching.
No.
So, and that I really like that you can really split your day up, like being in the lab and really doing things in the lab, but also like spread your knowledge, let's say, or
and
spread knowledge.
With the students as
indeed.
Well.
So that would be an ideal day.
All right. And do you think then that being an assistant as well, that you gain additional competences by combining your PhD with being
Definitely, because in our group we give quite a lot of practical sessions. So, which means that it's sometimes also really hard in managing, like when that you can do experiments, because some experiments cannot be done on half a day, so you need a full day. So I think it needs a bit more of planning than if you're just conducting research, that you're a bit more like, let's do this today, or I can check this today or something. And if I need to give class in the afternoon, I cannot just start up something which just popped into my head or something.
So at least planning skills are
yes, so
nurtured.
Planning skills indeed.
Yeah, all right. What advice would you give to fellow PhD researchers at the beginning of their career?
Just remember why you started it in the first place. Like, what do you like about research? Know that it's also a hard job, let's say, so that so some days are longer, some days are shorter. So just for yourself, that you need to, how you say it, plan your days accordingly. That if you have a long day, that you don't need to be the next day there from 7 till 7 or something, but that you just make sure that you need to do what needs to be done. And my advice is that to students that start, like, if you're not doing anything at the lab, don't just stay at the lab for the hours that you'll be there, but
make them count.
Make the hours that you're doing research count. And if you're in an off day or it doesn't come, then don't waste time on that or something.
Well, thank you for your time and good luck with your research and your presentation.
Yes, thank you.
Thank you.